darn it. And when it comes to the Punnett square, we use the Punnett square to determine the possible genetic outcomes of an offspring. And when we do this, we see that the alleles or the forms of genes, which we've been using as different letters, are either going to be heterozygous or homozygous. If they are different, one's capital, one's lowercase, that's heterozygous. If they're the same, either both capitalized or both lowercase, then that is what we call homozygous. Now, sometimes, again, if it's capital, it's dominant. If it's lowercase, it's recessive. We also looked at genotype versus phenotype, and a lot of your exam review worksheet has you practice knowing the difference between them. So genotype is a gene combination. And again, we've been doing this by looking at, we're representing them with the letters. Phenotype is the physical traits that are expressed. So a capital B, lowercase b might give an organism black fur. We then looked and defined genes, which we know are just a DNA that codes for a trait that can be passed down. Alleles is a form of a gene, and we've been using these as different letters. And then a ribosome is the actual organelle that will read the messenger RNA to make that protein. We're going to actually go through this process today, okay? But before we do that, a little reminder of how these guys are related. Genes have the information, they give that to the ribosome to make the protein, and then that protein is what gives you your traits, okay? Questions on this? Okay. So now we are going to add to our knowledge. So now is the time to add to your notes. I have a little graphic here, and we have the DNA. We see that transcription happens in the nucleus, and it gives us our messenger RNA. Notice the difference. I want you to sort of look at this and try to figure it out on your own first. And then it goes through translation at the ribosome to create the protein. This is the protein, and this green thing would be the ribosome, okay? Armand, is that a question or an answer?
I have a question.
Go for it.
So if the ribosome the RNA detaches from the DNA, where does the extra mass for the RNA come from? Does the DNA lose some of its mass?
What do you mean?
Like when the RNA separates from the DNA, the DNA stays the same.
Okay, so let me clarify what happens here, okay? In transcription, I was gonna let you try to figure it out, but I think it's better if I guide you a little bit. So transcription, we're gonna end up making a copy of only half of the DNA. So this is not a piece that has left DNA. It's actually like a photocopy. There are certain enzymes or proteins in the nucleus that will kind of unwind a portion of the DNA that it needs, kind of expose it, and then they make a direct copy. And that copy is this messenger RNA. There it is this messenger RNA. So DNA stays the same. After that copying system is done, DNA will come back together and like fuse back together and go back to normal. It's kind of cool. Okay. So, did you have a question, Sahil?
I don't know if I should ask it because I did some extra reading because I was curious, and then.
Okay, then hold on. Okay. Go to your formative now, and sort of based on what you heard from me, and just based on these pictures, I just want you to sort of think through. I already gave away what transcription is, right? Transcription is when we take a copy. We make a copy, a photocopy. Just like sometimes we make worksheets here at school and we make copies of them. That's what transcription is. So go ahead on formative. What do you think translation is then? Because I already gave you the first one, but you can fill in the first one too. Okay. This is wild guess time. You have no idea? Meh. Do give me an educated guess. That's perfectly okay. All right, so we're back on the formative. I have a couple of you sleeping on me. Izzy, are you back with us yet? Izzy, are you with us? Okay, I'm going to go ahead and mark you absent, Isabel. I'm sorry, Izzy. All right. Volunteers, if you already know, because you already looked it up, let's wait. But if you, I'm going to hear some of these wild guesses. What do you think transcription is, which you can just reinstate what I just said, or what's translation? What do you think these are? This takes a little bit of bravery. All right, I have some brave souls. Natalie, what do you think?
I think translation maybe means to move protein to another cell.
Thank you. What do you think, Armand?
Maybe translation is the transformation from RNA into protein.
Okay. Sahel.
I would say that which one? I'll do translation. I'd say translation is the turning of the photocopied instructions into actual proteins.
Okay, so when we look at this, we are going to actually see that transcription is when one side of the DNA code is copied or transcribed, right? If something transcribes something, they like make a copy of it to make messenger RNA in the nucleus. So DNA is stuck in the nucleus. It can't get out. It is such a massive biomolecule. It does not leave the nucleus. It's trapped. So in order for us to get the information from DNA out, we need to make this small RNA strand. It's really small. It's even half of it's not even a full double helix, right? It's a single helix. And it's going to be super short because it's only for the trait you want. And now that messenger RNA can leave the nucleus, and it's going to be translated now. So translation is when the ribosome translates or uses that code to make the protein you want. Yes, Armand.
DNA has mass, right?
Yes.
And then same with RNA.
Mass, M-A-S-S?
Yeah.
Yes.
So when RNA is created, where does that mass come from?
Okay. So when we are pulling things out in the cytoplasm, it's kind of like filled with all this recycled parts because your cells are constantly breaking things down and rebuilding them. It's really amazing what kind of factory our cells are. And so they're pulling the stuff out of the cytoplasm. There's specialized enzymes in all of your cells. And it blows my mind as I'm saying this, how tiny the cells are, yet how complicated and how intrinsic the system is. It's really, really amazing. Yes, Sahel.
So the nucleus is packed really tight, so they need to turn the DNA into RNA so that it can escape. But somehow they have enough space to pull the stuff from the cytoplasm.
Yep.
Crazy.
There's enough space for another molecule to go into DNA, unravel it, cut it, and then glue it back together.
It's kind of crazy.
Very crazy. Others.
So there just happens to be enzymes floating around in the cytoplasm.
Yep. Yep. It's amazing design, right? It's an amazing design. So
Just last question, then we can move on. Sorry. It's just I was wondering. So most of the ribosomes are in the rough endo something something, and then plants have a smooth one. But are there also some ribosomes just floating around, or like where are all the ribosomes?
there are a vast majority of your ribosomes are going to be found on the rough endoplasmic reticulum, the rough ER. Biologists like to simplify it. So yes, a vast majority are going to be found in like that ribbon-like structure. They're there, but there are ribosomes floating around in the cytoplasm as well. Yep.
Cool.
Others. You know what I'll do between now and Monday? I'll try to find, there's some really cool videos to kind of show you what happens. So let me write that down. Other questions? Okay, so we're going to pause here. When we get back, we're going to now focus in on that mutation because sometimes errors occur or changes occur in that DNA code. And we want to know, how does that affect an organism? Okay, so let's go ahead. We have 3 minutes. Get up, get a stretch, restroom. All right, come on back. Oops. Thank you, Angelica. Sahil, let me get through mutations and I will address all your questions, okay? So let's look now more specifically at mutations. So there's various effects of mutations, okay? Mutations could be helpful. That's okay, Angel, welcome back. Okay. For instance, thank you, Bailey, for letting me know you're back. For instance, what if the DNA code said, make the bug have red spots? Okay. But then there was a change in the gene, the code, something happened to the DNA message where instead of it saying, make the bug have red spots, it accidentally said, make the bug have green spots. Okay. So on the formative, thank you, Donovan. Thank you, Drew, for letting me know you're back. On the formative, go ahead and answer the question, how would this be helpful to the organism? So this is number 3. Bailey, Faith, Drew, Ayan, Caleb, Rehan, make sure you answer number 2. We went through that now. Number 2. We're on number 3 now on the formative. Okay. Try not to go ahead. I know some of you are anxious to get that review worksheet done. So now, why would green spots be an improvement, or why would it be an helpful mutation for an insect? So this is number 3. Okay. If you're stuck on number 2, come back to it later. Let's go to number 3 now. Okay. All right, volunteers, what do you think? Why would this mutation be helpful? Bella, what do you think?
This can be helpful because it's more common, or maybe has something to do with camouflage or something.
Okay. Matthew.
Maybe it might be an improvement because, like, for some animals, like, green spots mean like poison, so it might like warn off other animals that it could be poisonous, so they might not want to like eat it.
Okay. Armand.
Green spots might enable the insect to camouflage among plants to hide from its predators or prey.
Yeah, right. So those green spots are more likely going to help it survive. It could camouflage, it could hide. Maybe it's scares off predators. It thinks it's extra poisonous. So a mutation could be helpful for an organism, okay? It could also be harmful. So mutations could be helpful. They could be harmful. For instance, what if the DNA code was supposed to say, make large claws? But instead, the DNA code changed to say, make large paws. What do you think? How would this be harmful for an organism? instead of make large claws, now they're going to have large paws. Abigail, what do you think?
Since they don't have large claws and they have large paws instead, they won't have the claws to defend themselves against predators.
Okay, good. Armand, what do you think?
I have a question.
Yep.
So, where would we answer this on the formative?
Oh, this one doesn't have one.
Oh,
I sort of 50 time-wise. Sahil. No. Sahil, can you hear me?
Yeah,
Okay, go.
it cut out for a second. Maybe the large paws makes it more clumsy, so it falls over more.
Okay. So claws, I like how Abigail says, well, maybe it needs those to protect itself. Now you don't have that protection. Maybe it's going to mess up your balance. What else, Armand?
maybe it could get kicked out of its organism group.
Oh, shunned, kicked out, right? Possibly. Also, if you rely on those claws to catch your food, man, you're going to go hungry. So, right, if it's just going to be like little puff balls trying to catch your food instead of those claws that can really hold on to it. So, definitely could be harmful if we have this kind of mutation. Mutations can also be neutral, though. Yes, but what if so the question was, but don't paws have claws? Yes. But what if the code said instead of make large claws, they made large paws? So the paw got bigger, but the claw stayed the same size. Now you have big old fat paws right? It's going to be a lot harder to move around. So mutations can be helpful, they can be harmful, they could be neutral. For instance, the DNA code be like, make a gray nose, but there's a change in the DNA somewhere, and instead of saying gray nose, they say make a gray nose. It doesn't affect it, right? Yeah, we had a change in the message, gray with an A versus gray with an E. And don't ask me, I have no idea what the difference is. I'm sure you can Google it. But, right, they're both gray. And so it's not going to affect the organism either way. Okay. Now, okay, British, whatever. And so we have here a mutation. So we want to definitely have this definition in our notes, right? And a mutation is going to be a change in a gene or a change in the DNA of an organism, right? But we're specifically talking about DNA that codes for a trait. Remember, that's what the difference is between genes and DNA, that genes code for a specific trait. So if you have a change within that gene, that's a mutation. Now, this is important because remember that gene is going to code for that trait, which is coming from your proteins. Remember the proteins that are created. We have that DNA code. If that code changes at all, when messenger RNA gets copied, it's going to be wrong. It's going to go to the ribosome. The ribosome's going to then start making that protein or that molecule that's going to eventually give you your traits. Okay. Questions on this? Okay, go faster or go slower? All right, I had two of you say go faster. Okay. So on the formative now, just to recap, what are the 3 ways mutations can affect an organism? Go ahead and fill this in on the formative, and then we are done. What's today? Thursday? All right, let me check you on formative. Volunteers, when you volunteer, give me one way that you are here. So Izzy, I see that you have your camera on now. If you can hear me, we're filling in the go formative. Okay. And Randy, Bailey, and Rihan, go ahead and answer number 4. All right, give me one. Just give me one. There's 3, so we can spread the wealth. And then we'll let you get started on that exam review worksheet, which Garika, slow down. Okay. All right, Sahil, give me one.
It can be a negative change.
Okay. Negative. Could
Do
be.
you want an example?
No, we're good. Thank you. Derek? Derek, we can't
Sorry,
hear
I thought I unmuted. They can be a positive change.
Positive change. Good. It could be helpful. And Garika.
It can be neutral, meaning it doesn't do anything.
Okay, good. So could be helpful, could be harmful, could be neutral for this one here. Okay. Any questions for me? I should recap that slide again. All right. Round of applause. You guys officially know all things you need to know for genetics. Okay. So what we're going to do now, it's 10:20. We're going to let's take our break a little bit early, because then you'll have the rest of the class period to work on this exam review worksheet. So get up, use the restroom, get a snack, whatever. And then we have this one here. Yes, Armand?
Are mutations when the RNA isn't copied exactly or when the ribosome misinterprets the RNA?
Those could happen also. For 8th grade purposes, we're going to just focus on sometimes the DNA itself the message from DNA to RNA can differ. Sometimes DNA can change. The actual DNA in your cells can mutate. And that's a mutation.
But can it ever happen where the ribosome misinterprets the RNA?
Yes, if there is a malfunction in your ribosomes, there is, I believe, a specific disease where your ribosomes don't function fully. That is possible also. Okay. Others. Sahil.
So I said I did the reading ahead, so this wasn't covered in the topics, but I may have gotten curious. So I read a scientific article and then I got confused. So, so it uses the mRNA, right? And then the mRNA, I saw this little diagram that it uses a tRNA that are linked to the amino acids to fit it. If it fits and then it makes that connection, then it takes that amino acid and fits it. But I was wondering, does it just trial and error trying to fit tRNA with mRNA, or is it just somehow no?
They're like puzzle pieces. Remember we talked about like Legos almost? Like, so, but it's more intricate than that. Like they have special shapes and designs for them to actually blend together. Now, usually they're in the same area. They can easily find each other. It doesn't take that long. Other times it might take a while for them to find the right match. So it is sort of trial and error, but again, a lot of your ribosomes and all the supplies are going to be around that endoplasmic reticulum.
Yeah. So it tries fitting multiple until it finds the right one.
Yes, it's not going to connect until they are the right ones.
Okay.
Others. All right, sorry guys. Start your break. Ready? 3 minutes.